So right. Next. So let us see the nucleus. Here, this is a this is for your understanding. Okay, the structure of nucleus. Next, inside the nucleus, there are some chemicals. These are called as nucleic acids. Nucleic acids. So these nucleic acids, the monomers are called nucleotides. Monomers of these nucleic acids are called nucleotides. Nucleotides. There are two types of nucleic acids. They are DNA and RNA. DNA and RNA. So nucleic acids are of two types. They are DNA and RNA. DNA means deoxyribonucleic acid. Deoxyribonucleic acid. RNA means ribonucleic acid. Ribonucleic acids. And these nucleic acids are made up of the nucleotides. The monomers of these nucleic acids are called nucleotides. So DNA is made up of deoxyribonucleotide. RNA is made up of ribonucleotide. Ribonucleotides. Right. So if you observe the composition of these nucleotides, so these nucleotides, they are made up of nitrogen bases, sugar and phosphate. This you have to remember. The nucleotides are made up of nitrogen bases, sugar and phosphate. These nucleotides are linked by phosphodiester bonds. Phosphodiester bonds. The nucleotides are linked by phosphodiester bonds. Suppose say this is a one nucleotide. These are all linked by phosphodiester bonds. So the nucleic acids are made up of nucleotides. The composition of nucleotide is nitrogen base, sugar and phosphate. So nitrogen base, the name itself indicating they are basic in nature and they contain nitrogenous compounds, sugar and phosphate. Let us see one by one. Let us see the nitrogen bases. Nitrogen bases. There are two types of nitrogen bases. They are purines and pyrimidines. Purines and pyrimidines. Purines. So purines means they resemble a substance called puron. That's why they are called purines. Just it is for your understanding. Nitrogen bases, they are basic in nature. They contain nitrogenous compound. There are two types, purines and pyrimidines. Purines, they resemble a substance called puron. That's why they are called as purines. The purines are again two types. Adenine represented by capital A and guanine represented by capital G. Then pyrimidines are three types. They are cytosine represented by capital C, thymine represented by capital T, and uracil represented by capital U. Capital U. So nitrogen bases are purines and pyrimidine. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. Cytosine, thymine and uracil. Right? Next, let us see the nitrogen bases present in DNA. Nitrogen bases present in DNA. The nitrogen bases present in DNA are adenine, guanine, cytosine, and thymine. That means A, G, C and T. These are the nitrogen bases present in DNA. And the nitrogen bases present in RNA. Adenine, guanine, cytosine, and uracil, U. So you have to remember here. So this adenine, guanine and cytosine. Adenine, guanine and cytosine. These three are common in both DNA and RNA. You have to remember the thymine is present only in DNA and uracil is present only in RNA. This you have to remember. So nitrogen bases are of two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. So this adenine, guanine, and cytosine. These are common in both DNA and RNA. The thymine is present only in DNA. Uracil is present only in RNA. This you have to remember. Okay. So the nitrogen bases present in both DNA and RNA are adenine and guanine, cytosine. The nitrogen base present only in DNA, that is thymine. The nitrogen base present only in RNA, uracil. This you have to remember. You can expect the question from here. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. Thymine is present only in DNA. Uracil is present only in RNA. Over? Right. Next. Sugar. Here, the sugar is pentose sugar. Pentose sugar means it is a pentose sugar. That is, it contains 5 carbons. 5 carbon atoms are present. 5 carbon containing sugar is called as a pentose sugar. Actually, the sugar is ribose. Ribose in case of RNA, whereas deoxyribose in case of DNA. So the sugar is ribose. Ribose is pentose sugar. Suppose say if you observe the structure of this is for your understanding. No need to remember the structure. This is for your understanding only. Suppose say this is ribose sugar. It is a 5 carbon containing sugar. First carbon, second carbon, third carbon, fourth carbon, and this is fifth carbon. Got it? Right here, H, OH, H, OH, H, OH, H, CH2OH. This is ribose sugar. Okay. Whereas deoxyribose sugar, it is also pentose sugar. That means it also contains 5 carbons. First, second, third, fourth and fifth carbon. Here H, OH, H, OH, H, CH2OH. This is the structure of deoxyribose. In deoxyribose sugar, here in the second position, oxygen is less. Here in second position, OH is present. Okay, hydroxyl group is present. Here one oxygen is less. deoxy. Deoxy means removal of oxygen. That is the difference here. That you have to remember. Okay. So sugar, it is pentose sugar, 5 carbon condition. Ribose is RNA. Okay. Ribose is present in RNA. Deoxyribose is present in DNA. Okay. Or in ribose sugar, it is a normal pentose sugar. Whereas in deoxyribose sugar, one oxygen atom is less. That's why it is called deoxy. Right. And next to phosphate. Phosphate is common in both DNA and RNA. So that is the structure. That is DNA and RNA. Okay. Right. And next. So over. If you observe the structure of the DNA, structure of DNA. If you observe the structure of the DNA structure was proposed by Watson and Crick. Watson and Crick in the year 1953. So according to them, the DNA is having double helical structure. Double helical structure. DNA is having double helical structure, or you can say it is double helix. Double helix, or you can say double helical structure. So like this. It is made up of two strands, two polynucleotide strands. It is one strand, it is another strand. Two thread-like structures. They are coiled together like this. Coiled together. Okay. So this is a helix. So this is helix. In this helix, the nitrogen bases are arranged like this. The nitrogen bases are arranged like this. A double bond T, G triple bond C, T double bond A. So C triple bond G, like this. So this is called as a helix. This is called helix. Okay. So the diameter of this helix, diameter of helix is 20 angstrom units. So each helix contains 10 pairs of nitrogen bases. 10 pairs of nitrogen bases. So this is helix, diameter of the helix. Each helix contains 10 pairs of nitrogen bases. That you have to remember. According to Watson and Crick, adenine pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds, 3 hydrogen bonds. You remember this one. According to Watson and Crick, adenine pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds. These points you have to remember. This is the structure proposed by Watson and Crick. So according to Watson and Crick, the DNA structure is double helical in nature, or it is double helix. Each helix contains nearly 10 pairs of nitrogen bases, 10 pairs of nitrogen bases, and its diameter is 20 angstrom units, 20 angstrom units. So according to Watson and Crick, adenine pairs with thymine by 2 bonds, guanine pairs with cytosine by 3 bonds, 3 bonds. And another point you have to remember, Chargaff rule. A plus G is equals to C plus T. That means 50-50. 50-50. So purines and pyrimidines are in 1:1 ratio. Adenine plus guanine. Suppose if adenine is 20%, the guanine is 30%. Cytosine is 20%, cytosine is 30%, thymine is 20%, like that. Okay. So this is the structure of DNA. To here clear now? Right. Next, RNA. Let us see the structure of RNA. RNA. It is called ribonucleic acid. Ribonucleic acid. Ribonucleic acid. It is made up of nitrogen base, adenine, guanine, cytosine, and uracil. So sugar is ribose sugar. ribose sugar. It contains ribose sugar. So ribonucleic acids. There are three types of RNAs. One is mRNA, and second one is rRNA, and the third one is tRNA. So mRNA is called messenger RNA. mRNA is called messenger RNA. rRNA is called ribosomal RNA, and tRNA is called transfer RNA. Transfer RNA. So RNA means ribonucleic acid. It is single stranded. Whereas DNA is double stranded. So RNA, it contains adenine, guanine, cytosine and uracil nitrogen bases and ribose sugar. There are three types of RNAs: mRNA, rRNA and tRNA. mRNA means messenger RNA. rRNA means ribosomal RNA. tRNA means transfer RNA. Just to remember the names, rRNA, ribosomal RNA. It is useful for construction of the ribosomes. It involves in the structural organization of the ribosomes. Ribosomal RNA. It is useful for construction of the RNA. Construction of the ribosomal RNA. Next, tRNA, transfer RNA. It transfers the amino acids required for protein synthesis. Transfer RNA. It carries the amino acids required for protein synthesis. Then mRNA, messenger RNA. So actually, listen. Say this is DNA. In this DNA, the genetic information is stored. So this genetic information is carried to mRNA. And from mRNA, this information is passed on to the proteins. That means the information present in the DNA will be expressed in the form of proteins via mRNA. That means the genetic information present in the DNA will be carried to mRNA. From mRNA again it will be expressed as proteins. That means the DNA is giving orders, and these orders are carried by mRNA, and these orders will be implemented in the form of proteins. Got it? And this process of formation of mRNA from DNA, it is called transcription. You have to remember this terminology. The formation of mRNA from DNA is called transcription, and the formation of proteins from mRNA, it is called translation. So here the mRNA is mediating the information. mRNA is mediating between DNA and proteins. So it is called as a messenger RNA. Right? Next you remember the genetic code. In mRNA, the genetic information. In mRNA, the genetic information is stored. In mRNA, the genetic information is stored in the form of codons, or in the form of triplet codes, or it is also called as genetic code. So in mRNA, the genetic information from DNA will be stored in the form of codons, triplet code or genetic code. Okay. So that means, what is genetic code, or what is codon, what is triplet code? A sequence of 3 nitrogen bases. A sequence of 3 nitrogen bases. A sequence of 3 nitrogen bases is called as a codon or triplet code or genetic code. So how many nitrogen bases are present in mRNA? How many nitrogen bases are present in mRNA? Adenine, guanine, cytosine and uracil. How many nitrogen bases? 4 nitrogen bases are there. How many nitrogen bases? 4 nitrogen bases. So these 4 nitrogen bases are arranged in the form of a set of 3. A set of 3. So what will be the probability? What will be the number of combinations of these codons? What will be the number of combination of these 3 nitrogen bases? That is 4 power 3. That probability will be 4 power 3. That means 64 codons are possible. How many codons are possible? 64 codons. You have to remember that one. Among these 64 codons, 61 codons are functional. 61 are functional and 3 are non-functional. 3 codons are non-functional. And these 3 codons, they are called, stop codons, or they are also called as termination codons. And these stop codons or termination codons includes UAA, UAG and UGA. You have to add this one. Are you getting? So the information present in the DNA will be carried to mRNA, and from mRNA that information will be passed to the proteins. And the formation of this mRNA from DNA is called transcription, and the formation of proteins from mRNA is called as a translation. So in this mRNA, the genetic information will be stored in the form of a sequence of 3 nitrogen bases. And the sequence of 3 nitrogen bases, say for example AUG, a set of 3 nitrogen bases. Okay. So GUA, a set of 3 nitrogen bases. Okay. AUG, GUA, CUA, this is another set. Are you getting? Like that, a set of 3 nitrogen bases forms genetic code, and that genetic code is called as triplet code. Genetic code or codons or triplet code. Got it? So that means here totally 4 nitrogen bases are present. Okay, a set of 3 nitrogen bases are arranged as codon. So what is the probability? 4 power 3. How many number of combinations are possible? 4 power 3 combinations. That means 64 combinations are possible. Among these 64, 61 codons are functional. That means they carries the information for protein synthesis. The remaining 3 codons, they does not carry any information, and these codons are called stop codons or termination codons. And these codons includes UAA, UAG, UGA. You have to by head this one. You will get the question like this. So, which of the following combination represents stop codons? Or which of the following combination represents termination codons? Or these are also called as nonsense codons. Got it? Right. This is the structure of DNA. Okay. Structure of nucleus and genetic materials, DNA and RNA. AUG, methionine. AUG, it is called methionine. Well, no need, just you remember these points only. Right? Move on. Right. And next, let us differentiate these terminologies. Genome, chromosome, gene and DNA. First you differentiate these terminologies: genome, chromosome and DNA. What is genome? So genome is the haploid set of genetic material present in a cell. Suppose say you consider this is a cell and this cell contains nucleus. In this nucleus, the genetic material is present. So, the haploid set of genetic material. The haploid set of genetic material present in the cell. Present in the cell or organism. That is called as a genome. Or you can say the entire genetic material present in an organism. Suppose, say for example, if you consider the sperm. Sperm contains 23 chromosomes. That means the genome is present in the form of 23 chromosomes. That is haploid set. Haploid set means single set. So that is called as genome. Or simply you remember the genetic material present in an organism or haploid set of genetic material. Haploid set means single set. I'll let you know that one. And then what about chromosome? The genetic material, this genetic material exists as structures, exist as specific structures, and these specific structures are called chromosomes. Okay. So the genetic material, for example, humans have 46 chromosomes. 46 chromosomes. That 46 number, that chromosome number, that is this 46, like this. They acquire a specific structure. Actually, in the nucleus, they are irregular. Got it? This is in the form of genome or in the form of chromatin. Now, this chromatin during cell division, they acquire a shape, a particular structure. That particular shape or particular structure you call as chromosome. Got it? Right. Suppose, and what is gene? Suppose say this is a chromosome, this is chromosome. On this chromosome, some specific functional units are present. So like this. The functional unit of the chromosome. This is called as a gene. The functional unit of chromosomes. The functional unit of the genetic material or the functional unit of the chromosome, that is called as a gene. Gene. Then what about DNA? DNA is the genetic material. DNA is the genetic material or DNA is the raw material required for heredity. So that means the genome is mainly made up of DNA. The genome is made up of DNA. Chromosomes are made up of DNA. Genes are made up of DNA. So we can say that the DNA is the raw material. Suppose if you construct a building. So that building needs some raw materials. Okay, that raw material is the DNA. Okay, the design of the building may varies. Okay, the shape of the building may varies. The structure of the building may vary. The size of the building may vary. But the composition is same. Okay, whatever may be the organism, whether it is a human being, whether it is a plant, animal or bacteria, whatever it may be, the composition of the DNA is same. What is the composition? It contains nitrogen base. It contains sugar. It contains phosphate. But where is the difference? It varies in the size of the DNA, quantity of the DNA. Is it clear? The humans have huge amount of DNA. The humans have higher quantity of DNA. The bacteria, as it is a microorganism, it contains very little amount of DNA. That is the difference. Is it clear? But the composition of the DNA is one and same. The composition of the DNA in each and every organism, whether it is a plant, animal or microorganism or a human being, whatever it may be, the composition of the DNA is, it should contain nitrogen bases. What type of nitrogen bases? Again, adenine, guanine, cytosine and thymine. It should contain the sugar. What type of sugar? It is a pentose sugar, that too deoxyribose sugar. One oxygen atom is less. And the phosphate is common. Then where is the difference? It differs in the quantity of the DNA. Okay, if you consider the genome of the human beings, the genome of the human beings is 3 into 10 power 9 base pairs. So, the size of the DNA, the size of the genome is measured in base pairs. Base pairs means what? Adenine pairs with thymine. This is one pair, one base pair. Guanine pairs with cytosine by 3 bonds. This is another pair. Got it? Like that, these pairs may be repeated. How many times they are repeated? That represents its size. So, the genome of the human is 3 into 10 power 9 base pairs. 3 into 10 power 9 base pairs. So, that means 300 crores of nitrogen bases are present. Okay. So, that is, if you consider the bacteria, this is a human genome. This is human genome. If you consider the bacterial DNA, the bacteria contains E. coli. E. coli bacteria. If you consider the genome of E. coli bacteria, it is 4.2 into 10 power 3 base pairs. Or 4.2 kilo base pairs. Simply 4.2 into 10 power 3 base pairs. Got it? So, like that, the genome can be measured. So, this is the difference between genome, chromosome, gene and DNA. Genome is entire genetic material. Chromosome is this entire genetic material acquires the shape called chromosomes. Got it? And then what about gene? It is a part of the chromosome which is functional. And then what about DNA? DNA is the raw material which forms the genome. DNA is the raw material which forms the chromosome. DNA is the raw material which forms the genes. Right? Next. Let us see the chromosomes and their abnormalities. Next topic is chromosomes. If you consider the humans have 46 chromosomes. In humans, the chromosome number is 46. The number of chromosomes in humans are 46. And these 46 chromosomes are two types. They are of two types. They are autosomes. These are also called as vegetative chromosomes. And the second one is allosomes or sex chromosomes. Autosomes, they are called vegetative chromosomes. Allosomes, they are called sex chromosomes. So, autosomes, they are 44 in number. Allosomes, they are sex chromosome, they are 2. Again, these sex chromosomes are again two types, X and Y chromosomes. So, chromosomes are broadly divided into two types. They are autosomes. Autosomes means vegetative chromosome. Allosomes means sex chromosomes. Autosomes are 44 in number. Sex chromosomes are 2 in number, X and Y. Okay, right. If you consider the humans, that is males, distribution of chromosomes. Males have 44 autosomes plus XY allosomes, whereas the females have 44 autosomes plus XX allosomes. This is the distribution of chromosomes in males and females. So, here the autosomes are similar in both males and females. But here allosomes, XY in males, XX in females. The autosomes are responsible for vegetative characters, whereas allosomes are responsible for sexual characters. So, in males, XY combination represents masculine nature, whereas in females, XX combination represents feminine nature. Right? Next. Let us see chromosomal abnormalities. Actually, humans have 46 chromosomes this 46 chromosome exists as 23 pairs. 23 pairs. 46 chromosomes. Actually, we have 46 chromosomes, means 23 chromosomes came from father, 23 chromosomes came from a mother. They exist as 23 pairs. And this 23 pair condition, this pairing condition, this is called as a diploid. You have to remember, diploid nature. And this diploid nature is represented by 2n. So, 2n is equals to how much? 2n is equals to 46. 2n is equals to 46 or 23 pairs. 23 pairs. This is called diploid condition. Okay. And next, trisomy. Trisomy, you remember these terminologies: trisomy, monosomy, monosomy, nullisomy, trisomy. 2n plus 1. 2n plus 1 means how much? 47. So, 2n is 46 plus 1, 47. Monosomy, 2n minus 1, that is 46 minus 1. So, 45. And then 2n minus 2, 46 minus 2, that is 44 condition. So, these are the abnormalities. You have to remember this one. So, total number of chromosomes are 46. They exist as 23 pairs. They exist as 23 pairs. And this 23 pair you called as diploid condition. The diploid is represented by 2n. This is normal condition. So, 2n is equals to 46, that is equals to 23 pairs. That is a normal condition. Suppose if one extra chromosome is added, that condition is called a trisomy condition. So, total number of chromosomes become 47. Monosomy, 2n minus 1. One chromosome is removed, deleted. So, then it becomes 45. That is monosomy condition. Nullisomy, 2n minus 2. Two chromosomes are removed. So, that is it becomes 44. That is a nullisomic condition. Okay. Alright. Let us see. Actually, I told you the chromosomes are two types. So, these two types of chromosomes are autosomes and allosomes. So, the abnormalities may be autosomal abnormality and allosomal abnormality. Let us see autosomal abnormalities. Autosomal abnormalities. Autosomal abnormalities. Example, Down's syndrome, Down's syndrome, Edwards syndrome, Edwards syndrome, Patau's syndrome, Cry-Do-Chat. Patau's syndrome. Next, Cry-Do-Chat syndrome. Cry-Do-Chat syndrome. These are autosomal abnormalities. Next, allosomal abnormalities. Allosomal abnormalities. Example, Klinefelter's syndrome, Klinefelter's syndrome, Turner's syndrome, Turner's syndrome, and Nullisomy. Nullisomy. So, these are the examples of autosomal abnormalities. Autosomal abnormalities. or right. So 46, that is equal to 23 pairs. It is diploid condition, it is the normal way. So if one chromosome is added, trisomy condition, 47 chromosome. One chromosome is removed, 45 monosomy. Two chromosomes are removed, that is 44 nullisomy. You have to remember this one. Autosomal abnormalities, example Downs, Edwards, Patau's, Cry-to-Chat. Allosomal, Klinefelter's, Turner's and nullisomy. Let us see one by one briefly. First, let us see autosomal abnormalities. So, the autosomal abnormalities include Down syndrome. Down's syndrome. Down syndrome. Here, the number of chromosomes are 47. The normal chromosome number is 46, but here 47. Extra chromosome is on 21st pair. Extra chromosome is on 21st pair. 21st pair. 5 o'clock. Extra chromosome is on 21st pair. So this condition is called trisomy 21. Trisomy 21. So Down syndrome, what are the number of chromosomes? 47. Extra chromosome is on 21st pair. So here this condition is called trisomy 21. Suppose say this is 21st pair of chromosomes. Okay. Say this is 21st pair of chromosomes. Okay, here an extra chromosome is added like this. Extra chromosome is added like this. So due to the addition of this extra chromosome, they will get some abnormalities. So these children are mentally retarded, they are physically retarded. There is stunted growth, physically retarded. They have broad forehead, and they are physically and mentally retarded. This is Down syndrome. Next, so Edwards syndrome. Edwards syndrome. Edwards syndrome. Here also the number of chromosomes are 47. The extra chromosome is on 18th pair. Extra chromosome is on 18th pair. So it is called trisomy 18. Trisomy 18. Trisomy 18. Edwards syndrome, the number of chromosomes are 47. Extra chromosome is on 18th pair. So it is called trisomy 18. Trisomy 18. Next, Patau's syndrome. Patau's syndrome. So actually their lifespan is very less. Edwards syndrome, the lifespan is very less. The children may born with some embryonic deformities, embryonic abnormalities, rudimentary organs, like they born with some heart problems, kidney problems, lung problems. So these are called as Edwards syndrome. And next one is Patau's syndrome. Here also the number of chromosomes are 47. Number of chromosomes are 47. Extra chromosome is on 13th pair. Extra chromosome is on 13th pair. So it is called trisomy 13. Trisomy 13. Trisomy 13. Here also the lifespan is very less. They also born with some embryonic deformities. So they live for some days. Okay. Next, Cry-to-Chat syndrome. Cry-to-Chat syndrome. Here the chromosome number is normal, but the 5th chromosome is abnormal. 5th chromosome. Part is deleted. In 5th chromosome, part is deleted. A part of the 5th chromosome is deleted like this. A part of the 5th chromosome is deleted. So they born with vocal cord abnormalities. That means the sound of these babies resembles the sound of a crying cat. Cry do chat means crying cat. The sound or the voice of these babies resembles the voice of a crying cat, sound of a crying cat. Over. So these are autosomal abnormalities. And next, allosomal abnormalities. Allosomal abnormalities, that is a Klinefelter's syndrome. Klinefelter's syndrome. Klinefelter's syndrome. Here, the number of chromosomes are 47. Number of chromosomes are 47. So these are sterile males. These are sterile males, means impotent males. These are sterile males or impotent males. So that means they have 44 plus XY. This is normal male. Normal male. Okay. Here in Klinefelter syndrome, 44 plus XY and another X chromosome is added here. An extra X chromosome is added. Are you getting? So 44 plus XY is normal male. Here to this 44 X plus XY normal male, an extra X chromosome is added. So he will get some feminine characters. So this is a Klinefelter syndrome. So these are sterile males. Sterile males. They develop some feminine characters. 44 plus XXY. These are called Klinefelter syndrome. This is Klinefelter's. These are Klinefelter syndrome. Okay, they will get feminine characters. They shows gynecomastia. Gynecomastia. So what is gynecomastia? Development of breast. Breast development in males. Gynecomastia. They shows gynecomastia. Gynecomastia means development of breast. So mainly you remember Klinefelter syndrome. What is the number of chromosomes? 47. These are sterile males. What is the genotype? XXY. XXY. 47. That is 44 plus XXY. Total 47. So these are called Klinefelter syndrome. So these are actually males, but they are sterile. They are impotent, and they show some feminine characters like gynecomastia. Gynecomastia means development of breast. So the sex organs may be rudimentary. And next, not transgenders. Transgenders are different. Not transgenders. Not transgenders. So they resemble some feminine characters. So we can say that means these Klinefelter syndromes, they may suffer with some physiological abnormalities also. Okay, not only the physical characters, they also suffer with some physiological abnormalities. So we can call them as Klinefelters only. Is it clear? The transgenders are different. The transgenders may be due to abnormality of the hormones, due to hormonal imbalance. Okay, they express some feminine characters. The male becomes female. Okay, the male undergoes surgery and they become a female. That is called transgenders. Is it clear? That is different. Okay. First you remember that one. And next, Turner's syndrome. Turner's syndrome. Here the number of chromosomes are 45. 45. These are sterile females. Sterile females. Their genotype is 44 plus X0. 44 plus X0. Actually 44 plus XX, that is female. 44 plus XX is female. But here one X chromosome is less. 44 plus X0. So these are sterile females. These are impotent females. There is no proper development of secondary sexual characters. This is Turner syndrome. Next, nullisomy. Just to remember the genotype, nullisomy, they have 44 chromosomes. 44 chromosomes. The genotype here is 44 plus 00. So these may be males or females, but they have rudimentary sex organs. They are all impotent. Rudimentary sex organs, impotent. Nullisomy 44 plus 00. Right? So these are chromosomal abnormalities. Next. So you remember another one, that is sex linked inheritance. Sex linked inheritance. Sex linked inheritance. So what is sex linked inheritance? Suppose say this is X chromosome, say this is Y chromosome, X chromosome and Y chromosome, and these X and Y chromosomes have some genes, on this, like this. Okay. If the gene located on this X chromosome or if the gene present on this Y chromosome are abnormal, and if they are inherited to the next generation, if they are inherited to the children's or offsprings, then it is called sex-linked inheritance. If the gene located on X chromosome is abnormal, if it is inherited, then it is called X-linked inheritance. X-linked inheritance. If the gene located on Y chromosome is abnormal, and if it is inherited, then it is called Y-linked inheritance. Y-linked inheritance. So just you remember the examples of this X-linked and Y-linked inheritances. Examples of this X-linked inheritance. Hemophilia. Hemophilia. Sickle cell anemia. Sickle cell anemia. Hemophilia. Sickle cell anemia. Color blindness. Color blindness, albinism. These are the examples of X-linked inheritance. Hemophilia, sickle cell anemia, color blindness and albinism. And examples of Y-linked inheritance. Hypertrichosis, ichthyosis, porcupine man. Hypertrichosis, ichthyosis and porcupine man. porcupine man. You remember these examples. What is hemophilia? Delayed blood clotting. Okay, that you have to remember, that is most important. So here, hemophilia. Hemophilia means it is X-linked inheritance. It is a X-linked inheritance. It is a delayed blood clotting. Delayed blood clotting means the clotting time increases. Clotting time increases. Generally, here there is a prothrombin. There is a protein called prothrombin. The prothrombin get converted into thrombin by an enzyme called thrombokinase or thromboplastin. So the gene responsible for this thrombokinase is located on X chromosome. If this gene is abnormal, the thrombokinase will not release properly. If thrombokinase is not released properly, prothrombin will not be converted to thrombin. So the entire process will be delayed. So generally the clotting time, when we get injured, the blood has to clot within 5 to 8 minutes. But the clotting time will be increased. Okay, blood clotting time will be increased. It will be delayed. The delayed blood clotting is called as hemophilia. And this hemophilia, it is also called as a royal disease. Royal disease, or it is also called as bleeder's disease. You have to remember this one. Hemophilia is called royal disease, or hemophilia is called bleeder's disease. And here there are two types of hemophilias. Hemophilia A and hemophilia B. This hemophilia A is because of 8th factor, abnormality of 8th factor. Hemophilia B is because of 9th factor. Hemophilia A is because of 8th factor. There are 13 factors for blood clotting process. There are how many factors? 13 factors for blood clotting. Among these 13 factors, if 8th factor is abnormal, then it is called hemophilia A. If 9th factor is abnormal, then it is called hemophilia B. Remember this one. And then sickle cell anemia. Sickle cell anemia. Sickle cell anemia. So due to a small change, the RBC becomes sickle shaped. So actually the RBCs are biconcave. That means dumbbell shaped. The dumbbell shaped RBC becomes sickle shaped like this. So the dumbbell shaped RBC become sickle shaped. It leads to anemic condition. That is called sickle cell anemia. So hemophilia, it is delayed blood clotting. The clotting time increases here because of abnormal production of thrombokinase. The gene responsible for this thrombokinase is located on X chromosome. So it is called hemophilia, and it is of two types, hemophilia A and hemophilia B. Hemophilia A is due to 8th factor. Hemophilia B is due to abnormality of 9th factor. Then sickle cell anemia. Sickle cell anemia means due to a small change, due to a small mutation, that means due to a change in one amino acid position, the dumbbell shaped biconcave shaped RBC becomes sickle shaped. That is called sickle cell anemia. Okay, right. And next one is color blindness. Color blindness. Here color blindness means unable to differentiate the primary colors. Color blindness means unable to differentiate the primary colors, unable to identify the primary colors like red, green and blue. So red color blindness is called protanopia. Protanopia. Green color blindness, deuteranopia. And blue color blindness, tritanopia. Tritanopia. So this color blindness is due to abnormality of cone cells. Abnormality of cone cells in our eyes. So from these cone cells, iodopsin protein, iodopsin is released. So due to abnormality of cone cells, the production of iodopsin decreases. The production of iodopsin decreases. So unable to differentiate the primary colors like red, green and blue. The red color blindness is called protanopia, green color blindness is called deuteranopia, and the blue color blindness is called as a tritanopia. You remember this terminologies, okay? So albinism, albinism is unable to produce the melanin pigment. They are called albinos. So due to abnormality of the gene responsible for melanin production, the melanin will not be released properly. That is called as a albinism. And next, Y-linked inheritances. Hypertrichosis means generally the Y chromosomes are present in males. So these Y-linked inheritances can be observed mostly in the males. So that is hypertrichosis. Hypertrichosis means dense formation of hairs on the body. Hypertrichosis, dense formation of hairs on the body. And ichthyosis. Ichthyosis means the skin becomes scaly like fishes. The skin becomes scaly and dry. The dry and scaly nature of the skin is called as ichthyosis. And porcupine man, just to remember those terminals, that's enough. Porcupine man means in some males, the hairs are sharp and pointed like porcupine. So that's why they are called porcupine mans. So these are some important examples of Y-linked inheritances. Okay, Y-linked inheritances. Right. So we'll stop here. We'll continue with the next class. In tomorrow's class, I will conclude the biotechnology part. Okay. All right. And day after tomorrow, I'd like to take space tech part. Okay. In the last class, I'll take the miscellaneous topics like emerging technologies, ICT. Okay, and also some health related issues. Okay.